and welcome to our video series about communicating science. So it's still me, Maria, and Daria.
Yes.
Yeah. Hello. So today we're going to talk about figures and visual presentations, right?
Yes.
We have made a small presentations about things that we at least find helpful in order to communicate science visually. And there's a few tricks that make it easier. And I think in general, visuals are really, really important to conveying the message, and it helps people understand what you're talking about. I think in general, for most people, visuals help to sort of get the concept of things. And I mean, it's different for different people, but at least for me, visuals are really important.
I would agree to that.
Especially, I mean, if I read a science paper, I need the figures to understand usually, because numbers in text for me doesn't really tell me anything. I think it's really hard to see trends and patterns if you just read text.
Yeah, even visuals usually don't help me either. And really big chunks of text to keep the focus, and yeah.
So, and I also I think you can't stress enough that today people are so used to visual things. I mean, we are more and more I don't know what to say, but I mean, we're getting more and more used to getting information via videos or like images and so on. And I think also that since we're communicating science in different ways, not only by publishing papers, but also putting things online and on social media, it's more and more important to have great visuals in order to come across like this giant noise of papers and information out there.
Yeah, definitely. And we thought to basically couple all kinds of knowledge from, of course, our journal perspective, but also as we have been researchers, to give you insights, and we threw this experience in as well, and our knowledge about what you just mentioned, Maria, social media, and it all is super connected. And then I'm also working as a scientific illustrator, so I have knowledge from other people like scientists, but other types of clients about visuals as well. So we wanted to combine all of this in one video, so you get, you know, everything basically about figures and visuals.
Yeah. So maybe we should start with our sort of slideshow and see if we have anything useful to share. Which I definitely think.
Sharing my screen now. So I hope, do you see the screen?
Yes.
Okay, perfect.
Okay.
Yeah, so I just start and then we will switch throughout the presentation. So as Maria introduced to you already, it is really important to have proper figures as a scientist. And it is basically, your figures that you publish or that you expose are basically the business card for the science world. Science world can be academia, journals, social media, anything. How you show your science in form of figures and visuals is basically, you know, your reputation, how you're seen in this world. Your science is your name, and then how you appear and how, you know, serious or non-serious maybe you can be taken. So they are super, super important. And why is this? It's like one of the biggest reasons is because we learn way more efficiently with visuals. I think visuals. Because like from childhood on, like we learn with pictures and picture books, and it uses way less brain capacity. So if I give you an example, for example, here, you have a text, and you would have to read through the whole text, these 6-7 lines, and a lot of brain capacity would be used until you know what this text is about. If I show you this, bam, you know, it's about a red car. So you immediately get the essence of what this text is about. If you want to know a little bit more details, You then know, okay, this is a red car here. I'm interested in red cars. So what kind of model is it? You know, how many horsepower, la, la, la. Then you go into the text and you filter what you want to know. So it is way more efficient to learn in that way. As a little analogy, I always somehow like to use red car. I don't know why.
You're interested in red cars.
And somehow, like, a red car will also look different in my brain than in yours, Maria, and in anybody of yours. So yeah, I give the floor to Maria now for a couple of slides.
Yeah, okay. So I think that you should, as a scientist or anyone who's wanting to present something, you should really take the time to do your visual properly. So you and your science, it's easier to be remembered if you have strong images or you have something that is really easy to follow. I think that you have to present things so that people understand. So it should be clear. And it's more likely that you will get a grant or be published or get this extra attention if you have great visuals.
Yeah.
And also, yes, if you have something interesting that people can look at, you won't lose your audience as easily. I mean, if you give a talk, we've all been to talks and we have all sat in there. And even if the topic is really interesting, sometimes if there is just a lot of text on the slides and so on, and even if you find it interesting, it's sometimes really hard to keep focus. But if you have these like really strong images, it will draw you in a completely different way. And also if people have sort of small videos or clips or things, it is easier to follow and remember also.
Yeah.
And also if you have, I mean, especially if you have these days, like your social media, it's also a platform to find other people that are interested in the same things as you, and you can increase your network. And if you have great visuals and figures and things, people will be drawn to them.
Yes.
And connect.
Yes.
And then, of course, it's all about trying to get people to understand what you're trying to convey, the message, basically.
Yeah. Yes. And actually, I just, a thought came to my mind. I once talked with I think it was a neuroscientist, and he said that in the brain, there's only one channel for understanding. Like if you listen, you can listen and watch someone, but you can't at the same time read. So the more text you have, you know, there's only just this one channel in the brain that takes in this information. So you will lose people when you want them to read a lot of text instead of listening to you and just watch quickly and grasp everything. I thought it was interesting. Okay.
Yeah, I mean, I've never really thought about why this happens, but yeah, because it's so easy. If you start reading something, you don't hear, and then when you have read the thing and you start listening again, they're in a completely different place in the presentation. You're like, well, I lost.
Anatomically speaking, we are not made to do that. So
okay, it's not just me.
no, it's me for sure as well. So the next slide, yes.
Yeah. Okay. So there are some rules that is easy to follow, that you should follow for easy understanding.
Yes, and we always thought to give like 3 to 5 points or something. So
yeah. So if you're sending in figures or graphs or something to a journal, you should stick to the guidelines. And I mean, most journals nowadays, I mean, they don't really care about the first submission. I mean, you should definitely have figures that people are able to read. I mean, yes, they should be easy to understand and follow and so on. They should be correct. But when it comes to the final submissions, you should really try and read the journal's guidelines. This will save you time. And usually journals have thought about this. They don't give you guidelines just because. There is a purpose to this. Yes. And I mean, it's usually that they just want you to be clear, that people can understand the figures, be concise. You should just follow a structure that you have started so that all figures sort of are connected. use the same font, use a font that is easy to read. Don't, and you see,
I forgot to click the.
that's okay. But I mean, exactly as we said, that you shouldn't overcomplicate things. Don't throw in too much if it's not needed, because that will just draw attention away from your specific thing. And then, as I said, you should really try to be uniform. Use the same color scheme throughout. For instance, if you have a wild type and you give it the color, I don't know, blue, stick with blue. Keep it blue throughout all of your figures. Don't change and switch it up. The same if you have a mutant, use the same color for the mutant, so on. And also, please do not mix red and green. I mean, this is something that we come across all the time that people forget. And that's just because you want everyone to be able to read your figure. And there's quite a lot of people that are colorblind to different sort of things. That's why. So don't do it.
Oh, yeah.
Yeah. And then exactly, avoid beautifying without purpose. Usually don't do 3D graphs. They're just, I don't know, they're harder to read for people, and it doesn't give you anything extra. There is no win if it's not for a specific purpose. If it's just a regular bar graph, just do a regular bar graph. And also, this is my really thing that I really hate most above all. And especially nowadays where there is no fees for color printing, or there is nothing. Don't use this kind of hatched bars or dotted bars, or because it's really difficult to tell apart.
Yes.
I mean, in this case, for instance, it would have been better to have one that is white and one that is black, and then you have one that is gray and dark gray. Even that would have been better. Yeah. But today, almost all journals allow free color images because everything is online.
Yeah.
So.
Yeah. And also, if you look at the legend, then in the small little squares, you can't even distinguish what's what. So your scientific message might not at all come across on any level. So
no, then you're just hoping that the authors have put them in the right order, and then you go from there. But it's not a safe way to do science.
No, definitely no.
So don't do that.
No. I would actually agree. I also don't like it. And then we have split the presentation in different ways how to visualize your data. So it's pictures, graphs, models, and we thought to go through each one of them separately, and we made like a little do's and don'ts list, and then come up with examples of what we've been publishing in Physiologia Plantarum. So Maria will go, we'll tell you a little bit about the do's and don'ts, and I will go through the examples. So here we start with pictures, photographs, basically.
And I think that to begin with, when it comes to pictures, I mean, most people when they're submitting papers and so on with pictures, it's usually of experiments, either like setups, or it is of plates like with colonies and stuff, or it's plants, something like that. And I think that this is where you have to remember when you're doing your experiments that this is hopefully going to be published. So make sure you take nice pictures from the start, because it's, especially with plants, I mean, it's really hard to go back. If you have crappy pictures from the get-go, then that's what you have. And it's really hard to do something about. And then the journal will ask you like, can we have a nicer image or picture of this? And it's like, no. So you should really take care when you take pictures, and you set them up nicely with like a white or a neutral background, and you do take care. Just take pictures, and it's better to take more pictures than you think you will need, and really document, because this is part of the experiments and the setup. If you're going to show a phenotype or something, then so you should have high resolution pictures. And this is also, I think that some people, they do take nice pictures, but then they sort of try to mount them in some other, like in PowerPoint and something, and they save them in a lower resolution. And there are tips and tricks for how to avoid this degeneration of pictures.
Yes.
So before you send them into a journal, make sure that they are in high resolution. And if you don't know how, if you had a nice picture from the beginning and then it turned out crappy when you do a compiling of your pictures or something, and you don't know how to rectify this, you can always contact a journal. At least we do a lot of these. We help authors with this if it's a problem.
And also the chance that if you have a really nice picture that it's picked for a cover, for example, is then also quite high. This is something that when you work at the journal, we look at this like, oh my God, we mark them. Great figures, great pictures, potential cover pictures. So it's extra exposure for you then.
Exactly.
The next point.
Yeah, that's also like from what we said before, be clear and don't have too much. I mean, also it's the same thing when if you have like plants, you should perhaps just set them up nicely, no noisy background, maybe one plant at a time or like 3 or whatever, but take care. And then yes, you should not have, or maybe I should say it's better to be balanced and light and neutral colors and try and be not too messy. Don't have like super bright and high contrast colors and things. It's harder to work with. And as you said, no neon colors or like really.
Yeah, or sometimes actually once I had an example where people would have the authors would have plants, green leaves or something, red background.
Yes, I've seen that several times also. It's really hard. And that's hard to do something about enough. I mean, yes, you can use programs and change the backgrounds, but sometimes that's also difficult because we do all of these image checks, and these programs, they sort of light up or they give you a notification that this has been tampered with. And even if it's just changing the background, of course we can talk about it and it will probably be fine, but you will get this little warning sign on your manuscript.
Yes,
on these programs.
And
it's important to label things properly and also clearly. And sometimes people do label and they take a picture and then the label, I mean, it could be a written label on the pot or something, and then it's like really difficult to read or see, and then it's better to have it afterwards in the figure later on than having something scrambled, like a small little
so these were the do's and don'ts on the pictures and photographies, and now I just show you a couple of examples of what we published. So the first one you see here is just a close-up of the part of the plant that's flowering. So the labels are good, the background you see is neutral, you can clearly see the flowers, you can also clearly see the color changes, so there seems to be maybe something around anthocyanin accumulation. So everything is clearly visible, distinguishable, 3 different plants, all roughly in the same stage. So that's a really good example of where to look at.
And I think it's good that they have had sort of the same distance when they took so they are sort of
you don't it's all good there.
Uniform.
Another example that is like zoomed out on the macro scale, you can also show your experimental setup, like in that case. Then it's not like super detailed, but you just want to see, okay, this is how we grow our plants, again labeled well, clearly distinguished, and you see the differences in what is in the happening in the experimental setup. So this is usually also when you have a very data and graph heavy manuscript, where you don't have so many models, for example. It's always good to have some kind of visual, at least to show, okay, these are the plants that we worked with and this is the experimental setup, and then we derive everything from our experiments in form of graphs. It's easier to publish something like this on social media than a graph
Yeah.
with white bars. It's
I was just going
easier.
to, okay, I'm sorry. Yeah, I was just going to say that even if you don't end up using these pictures for the actual publication or for your article, it's really good to have for social media and stuff. And we get quite a lot of manuscripts from China using native plants, for instance, medicinal plants and so on, that I have no idea what they look like. So I always have to Google and look like, what is this plant?
Yes.
So, I mean, if we have something like you take a picture and then we can use it for social media, for instance, and because there will be more people like me that are not familiar with these.
Exactly. And it's super interesting to learn this and to see, like, not the typical model plants. And there's all kinds of plants that research is done on. So it's really good.
Yeah.
That gives you an exotic point. Next example here, in that case, the focus is on the fruit. So you don't have a black background because the fruit is growing on the tree. So they just wanted to show, I guess a certain type of mutant and a Y type, and just the development of the fruit and how it looks at different stages. And because it is so distinguishable, this red and this dark and the light, it doesn't matter so much that the background is not uniform. So there is cases where it's also okay. So it's not black and white, but you can have this as long as you make sure, label it properly and have it really clear, like focus on one cherry and not have a lot. And then the reader needs to look, where should I look at? It's very clear, bam, 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 these four. And and the last photograph is, like Maria mentioned in the beginning, sometimes it's plates as well. So in that case, they wanted to show how the plants grow over time, first in these sculpture glasses on plates, and then they are like in bigger plates, and they are transformed. So you could already guess that it's several different, days and maybe weeks that the plant has been growing, and still the background is black. So the authors really took care and put effort into showing what's the experimental setup to have it as clear as possible. So extracted most likely the single parts of the pictures and put it on a black background. Super great, because like this, immediately the experimental setup, you can see everything at once clearly, growth stages, and it gives the feeling that they cared about the readers actually understanding what they have done. That's a really big thing about having proper visuals. Like, psychologically speaking, it shows you care. You care about your science, and you care that others understand it. Huge plus point.
Yes.
So that's for the photographies. Do you have any other inputs there, Maria?
No, I think that we have covered basically the things that are important. But it's usually, I mean, it will solve itself if you think beforehand.
Yeah, exactly. Yeah, I agree. So then we're coming to the graphs, because that's the other, it's a big part of scientific data, showing them in graphs. And we have, again, a little do's and don'ts list. Maria, should I show all of them at once? Is that easier?
Yeah, it's easier. Yeah.
5. And I'll go for it.
I mean, some of the things we have already talked about. I mean, like the grayscale of colors versus many patterns and 3D effects. I mean, stick to grayscale or colors. And if you do grayscale, include white and black so that you have a larger spread of things. And then don't do really busy graphs. Sometimes people tend to want to do everything in one, and they have trend lines and they have like a lot of things in one. And I think that's sometimes really confusing and hard. And if also some people try to have different scales and for me, I think it's hard, because then you really have to sit and look and try to, like, what are they trying to tell me? It's not like immediately clear. You have to read the legend really carefully sometimes. And then also try and have similar y-axes so that you can read across. Especially, I think, if you have different, you have different panels. This is something that people do sometimes, and they change the scale. I mean, sometimes you have to do it. But if you have things in a line and it's something that you think will be similar, and then you realize like, oh, but they've changed the scale here, so they look like similar, but actually, if you take the scale into it, I mean, this is super small compared to the first. So I think then you lose sort of the message that they are different. So, and then of course, background noise. Don't have I mean, for me, I mean, this might be different. I don't really enjoy when people put too many like lines in the background so that you're supposed to be able to compare. I mean, some fine, but a lot, like a typical Excel, like
yes,
I don't see the point most of the time. And then proper labels. And also remember to have statistics in a clear way. Not overdo it. I mean,
so these were the most important parts for graphs, and now we go into examples again. So first, a couple of examples how it should not be done, or we would say it's not ideal. Let's say it's of course you can do it, it's up to you, you know, it's free world, do whatever you want, but it like advisable to maybe refrain from it. And that's the thing that I mentioned before. And now you see it a little bit more zoomed in, all the patterns and everything. It's super noisy. Looking at it, I get dizzy. So this is really something to be avoided big time.
sure.
No, nothing is. Just skip this completely. Then here, it's just a really basic, you know, I typed in a couple of numbers in Excel. If I see this, I know, okay, the person just used Excel and didn't put any extra effort to make the data represent, like, you know, put extra effort in it. So I'm a little bit disappointed when I see this. You have these extra lines here and this typical color scheme, not even adjusted to anything else you might have in the manuscript. So it's a little bit of a, you know, just extracted it from Excel. Like, you put so much effort into your science, years of work and money, funding, people, stress, you know, disappointment until you get to the point of publishing it. And then it's just an Excel thingy. It doesn't represent, you know, that's a shitty business card. Oh, sorry, I shouldn't say that, but it's a not ideal business card. That's not the effort you put in. It's not these colors in the Excel thingy.
You can do that.
And
then we come to the goal.
That's what you mentioned before. Everything. It's, you know, a little bit of 3D effects even there. Lines, arrows, labelings. Nothing is possible to read in this. And that's, I guess, that would be intuitively for everybody like, hell no. So don't do this because it's really difficult to read. 3, you know, axes as well, everything, different colors. Impossible. And if you really look at it, it's still the basic Excel. It's still an Excel plot. But a lot of effort is put in, but maybe not in the
right.
No.
And I mean, this is just going to confuse people. And as you say, I think that I think it was a key thing that you said, that you put so much time and effort into and everything else, like designing your experiments, doing hours of like work. And then you sort of fall on the finish line.
Yeah, exactly.
Basically, because this will not help your science.
No, no, not at all. But of course, there's also, you know, there's ways to do it differently, and it can be very simple, like you see here. Boxes, black, white, gray. That's it. Statistics, labels, super clean, aligned as well, as good as possible. You have that's just simple without any fancy colors. If you don't need to use colors.
No, I think sometimes the simple figures are for me the best, but it's just you can immediately see when you open a manuscript and you're going to read it. You can immediately see that, oh, this is going to be something good, because I'm not saying science, the scientific part might be good, but you see that there's someone who has at least taken care.
Yeah.
They have tried to present their data in a nice, easy way.
Yes.
And I think for me, I mean, if I have done that, I would still be a little bit like the panel B is a little different in size than the other ones.
Yes.
That I might have changed if it was me.
Yes. And actually, you know, A, B, and then goes up C, D, and one is not labeled. So there you see, sometimes it happens, you know, you could
But
like,
I mean, still, I mean, this is so much more preferable even so. I mean,
yes. And you can say, and you can switch it around. It's not a problem.
No, I mean, this can easily work.
Yeah. And the next example is also a very simple one, but here like one color edit. It could make already a big difference. Like, you know, okay, highlight, grown in highlight. You have the yellow line here. And then you keep on using the same yellow throughout your figures and throughout your whole manuscript. So it makes it easier for the reader to follow everything that you're saying. So again, it's actually pleasant because you're taken by the hand, you know, like, look, through the complicated science and the years of stuff that I've been doing, this is what I found out. Amazing. What one color can do.
Exactly.
And then last but not least, sometimes, you know, we also have like figures that are very heavy in statistics, bioinformatics, and then they could become a little bit more complicated, but even that you can try to make it a little bit easier. In that case, you know, use like a color gradient to see like that's the distribution of the different sample sizes, and to really make sure that's the line, the dots, and then you have different shades of the same color that show you something different and maybe represent a statistically significant area. Sorry. So it is very simple tools to use a couple of colors in graphs to make a difference. Yeah. So this was all for graphs. Now I think we're switching back to not back, but to models and illustrations. General hypothesis, showing hypothesis and models. And Maria will again, I will just come up with
yeah, and I think in general, I mean, it's sort of the same rule that applies over and over again, that you should, if you just stick by these for basically all your visuals, you will do fine. I mean, you should just try and see how you can visualize this in a simple and clear way. Don't throw in too many things. Don't do too many colors, too many arrows, and keep it simple. And also, I think that you should try and see that you have consistent shapes. That's also one thing. I mean, that also goes for graphs. Keep the same sort of line, what you call thickness for everything, and so on, the same font size, and don't do bold in one and regular font in another. Like, just try and keep uniformity across all things, and that comes, it's the same thing for this. And I think also you should have in models and so on, you definitely can have text, but don't overdo it. I mean, it should help you, so that without text is also really hard, but too much will also confuse you.
Yeah.
And then, I mean, it's usually best if you try and do this on your own and not do something from the net.
Yeah.
I mean, they're great. They're great resources out there today that you can use parts of. I mean, you can get shapes, you can get everything that will help you to do something that you have made yourself with bits and pieces from the net. You don't have to be an artist to do a model or an illustration. That is
definitely not. And a lot of people feel actually shame about, like, most of people say, like, oh, I can't draw. Like, if you actually can draw, like, it's like the simplest thing is possible to draw shapes, but we'll get there with the examples. Yeah, so maybe I could actually now jump already to the examples. Models and illustrations. For example, here you see It could be a combination of having pictures, a little text, some small illustrations, expressing a high or up or down regulation. That could be just a very simple model at the end, like, okay, this is what we have found out.
Yeah, and I think in this case, I mean, they have a red and green, but you can still see by the shape of the arrow if it's going up or down. So I mean, even if you can't distinguish between red and green, I mean, you will get the idea from this one. So
exactly.
if I would have picked different colors, I mean, it's still clear.
Yes, and I think they might have thought about it because that green is a little bit more bluish green, so there are shades of
my screen.
Yeah. But ideally, yeah, should be avoided, as we said before. Then in that case, they included data actually in the model. So that's another option. You have this hierarchical cluster data of different metabolites, and then you just incorporate that into your model and illustration next to a plant that you've maybe drawn or taken as an icon. So it is possible to have this as a final model as well.
Yeah, and I think this is a great example of adding interest into one of these pathways. Because people, a lot of scientists use, like, they have an illustration of a pathway, but they don't connect it to a plant or to something. And it doesn't have to be a lot. I mean, they have a green background, and sometimes you can maybe just see, where is this? You can have it from a leaf or in a bubble. So it connects it to something, and it makes it more interesting than if you have just a white background and a pathway.
Yes.
That's, at least for me, I think that's a huge difference.
I think so too. It's just gives you the bigger context, you know. Sometimes people are scared about pathways and metabolites and all these arrows and cycles. They're like, oh my God, I don't know. But then connected to a plant, then it's already like you bring ease to their lives and their brains. And it's always next example here, because we were talking about self drawn before, and you see that the authors tried to draw the different cells of the parts of the maize leaf by themselves. This is awesome, amazing. It doesn't have to be like super perfect. This shows such great effort that they put into thinking about it and daring, you know, to submit their own hand-drawn cells. Great. It's really, really perfect. So no shame about that.
No. I think these are great examples of something that sort of just improves understanding and makes it interesting. And I think that if you take these things, because we try and advertise and try to get readers for all our published articles. And we go through the articles trying to find something, a figure or something that is nice to publish on our social media. And sometimes sometimes maybe you don't have anything that you find. I mean, you have graphs and they're okay. But if you have models or illustrations that stand out a bit, it is so much more effective.
Yes.
It will draw more attention to your. So even if you don't have like these things, having a graphical abstract, for instance, is something really great. And I think more scientists should do that.
Yes, you will immediately get attention, and everybody will know, oh, it's about nice plants. Something on a cellular level. Great, I'm interested in it or not. And then the likelihood that somebody, you know, catches your science, your article is way higher. Because again, we are super visual, like visual learners. Yeah, and I think we have one more example here. Yeah, where you see a little bit of mix again. They might have used maybe some icons from the net, but you know, with a little bit of labelings and something done themselves and shown a little bit of a, you know, pathway with explanations. It's similar to this one here, and a little bit of a, another way to, again, like have this, what Maria said before, you know, all this might be a bit abstract. Where does it even found? Oh, well, the context is, you know, winter, spring, so you have immediately something to connect to. Yeah, so this has been it for models and illustrations. And now the last one that we thought is also worth to show is combinations, because now we've gone through photographies, graphs, models, and it could also be that you can combine all of these into one figure or one model, and how that could look. And I mean, as you said before, it's most likely very similar to what we've already talked about.
Yeah. And I think also when you say here organized panels, messy panels, I think that that's something that some people, when they do their figures, don't think about. You should think about how an article is set up. I mean, usually you have 2 columns, and you can either have a figure that spans over 2 columns, or you can have 1 column. And in some cases, you're allowed also to have 1.5. So, but I think that you should always think about this before you start doing your figures, because then you can see how big will this be? Can I read it? Is it possible? Is it too small? Is it, I mean, and then adjust your panels and things to this size. Yeah. So maybe you can take up the next. I'm sorry, but I will have to connect my computer because my power is not running.
Yeah, no problem.
I will disappear under the desk for this.
No problem. I would just continue. And now we have this, color, proper labels, and the colors again. If you do like a combination figure, then meaningful colors are even more important, because everything is kind of different, and you want to make it even more easy to connect what should be connected. So we have 3 examples, and the first one, this one, is actually one of my most favorite. So you see here pictures, different types of leaves, different colors. You have here like a phylogenetic tree, and then you have data analysis like 2D, and here 3D, which we before said like maybe don't do it, but they have 3 different scales and dimensions, so they needed to show it in that way, but it's not as overwhelming because they used colors so smartly. They used it's amazing, the colors from the plants, they extracted it, used that in the tree and the graphs and in the 3D dimensional plot as well. So this is a really good example on how you could actually, when you use all of these things in a smart way, bam, you have this great figure.
They're sort of connecting the dots.
Yes.
They're using a color in a really smart way.
Yes, and yes, and it's green and a little bit like purple magenta, and that's recognizable for colorblind people as well. So they didn't pick the red, but they still wanted to represent the plants that they work on. So it's really good.
It's a great example.
Yeah, immediately clear. The next one is, in that case, we have plants watered, not watered, pictures, and then graphs. And even in these graphs, you have this little bit of an extra visual cue that you added that makes it all like really pleasant to look at. While I generally wouldn't recommend just beautify something for beautifying it, in that case, it's to me, it's understandable. They have a lot of different labelings to make it just a little bit more accessible to people, to still see like, okay, it's connected to a plant, to an actual leaf, so that people don't forget it. So it's a little bit of I don't see that so often.
No. I think this might be the first time I see that. Yeah. I think that this might not be for everyone.
No, I agree. People could be like, oh, what the heck is this? So yeah, but in that case, again.
But even so, I mean, they have done it in a very subtle way. And even if you're not a big fan, you can still easily read figure. Yes. And it's not, yeah.
Yeah. And I mean, I will remember this now. And if I'm interested in a topic, I remember the authors. So it could also be that a journal, you submit this and the journal says like, no, could you remove it? Then you remove it, but you can give it a try. If it's something is, subtle, it's worth to give it a shot. And then you just see. And the last one is this. It's pictures, graphs, and plates, all clear, and in one figure, done. And
Yeah. And I mean, also the layout. I mean, they have done it in a way that it's sort of uniform across. It's really easy. Yeah.
So I think this was actually, yes, all that we had. I will stop sharing.
Yeah.
Go back to Zoom. That's all we wanted to share with you. Anything else, Maria, that we forgot?
I think that it's the only thing I would say is that I think people should be proud of their work and do good figures. For me, it's like the one thing that stood out from this whole talk when you were talking, it was like the thing is that you do all of this and then you do a crappy figure. You've done years of work and then you did this. Why?
Yes.
And I mean, today, I mean, I can understand previously it was hard. I mean, trying to do figures in an easy, nice way. Today, there is no really excuse. I mean, there is open source things everywhere. You don't have to have a really expensive program or anything. I think today everyone can do a good figure. You just have to follow some really simple tips and tricks, and then everyone can do it.
Yes, very much.
Yeah, just give it a little bit of extra thought. And then also from our point of view, I mean, we try really as a journal to help people. I mean, we go through all published manuscripts and we give tips and tricks, and we try to help authors to get better figures and give them pointers to maybe you should change this, don't do this. I mean, I don't think I've ever said okay to a figure that have these like hatched parts and stuff. I always send them back and say, please, this is too hard. Don't do this. So we really try to help. And if you have problems with a figure, you can always contact us and we will help you. Give you tips and tricks if you're finding it hard. But also try and include something that is good for our social media platforms. Include a photograph or a model or anything you can think of that is a graphical abstract, because we will be really grateful and it will help spreading your science.
Yes,
it definitely will.
Yes, we put a lot of effort into reaching people on social media, so we have big networks, so it's possible then if you have a great visual and we put it out there, that somebody finds you that has not known about you before just because of this, and it's very powerful. So any effort in visuals and figures will never be wasted.
No.
And nobody should ever say that it's wasteful to spend time on properly depicting your science, because it never is.
No, and I think if you do great visuals to begin with, you will have use for them later on, because you are always going to go to conferences or give talks, or and then you already have nice visuals, something that is easy to use. You don't have to redo them or trying to get them suitable for a different format. It's already nice and clean and done.
Yes.
Yeah. Okay. Yeah. So as usual, we encourage you to get in contact with us, follow us on social media, comment, ask questions, whatever, and submit papers.
Yes, that as well. So, yeah.
And I guess we will be back with something. I don't remember what the next episode is about. Do you?
It will be actually about social media, but we will have a look. So, yeah.
Okay, so we will be back with an episode on social media.
Okay, perfect.
Okay.
Thanks a lot.
Bye-bye.
Bye.